Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on earth, and that's biology. So welcome to my new beginning series. Hi dear 𝗍𝐴. So thank you so much for joining me this evening. So please do like, share and subscribe. So here we are talking about biotechnology. So this is part 4. So we are doing it in very small bits and pieces. So today we are in part 4 of biotech. So where are we? So we're going to talk about vectors today. So we are done talking about how restriction enzyme works. What are the different types of restriction enzyme? How do you verify the cross verify the step that your restriction enzyme has worked or not with the help of gel electrophoresis? And from gel electrophoresis, how do you proceed to the next step, and that is elution. So till that is done. Yes, so today we are going to talk about what are the features of a vector and how do you cross verify a vector has ligated with the gene of interest. So these are the two concepts that we are going to talk today. Hi Natasha, yes, after a very long time you are here, so thank you so much for joining my session. So please do like, share and subscribe. So chalo, let's move forward. So I'll teach you only two concepts, so I'm doing it very slowly so that you understand the essence, and there is no point in hurrying, because this chapter gives you a lot of base, a lot of insights into your future scope, right? So let's move forward. So this is part 4. So here we are, if you remember, tools of recombinant DNA technology. So we are done with restriction enzyme. Today we are yet to talk about cloning vectors. So let's talk about cloning vectors, and for another day we'll keep competent host. So once again, what am I talking for all those who joined my section today? So let me tell you. So we are talking about the process of recombinant DNA technology. So how it is done? So first I find my gene of interest. So you were watching my recorded lectures, okay Natasha? So if you have any doubts, please get back to me. So I'm always ready to clear your doubts, okay? Right. So how do you do recombinant DNA technology? So from a particular DNA, I find my gene of interest, and I have to insert this gene of interest into another DNA. Right? So through a vector. So this is the concept that we are talking about. But from the entire DNA, for me to cut this gene of interest, I need molecular scissors. So that molecular scissors is known as restriction enzyme. So once I have my restriction enzyme subjected to the gene of interest, how do I know this has chopped this portion? Because everything is microscopic, I cannot see anything, right? So how do I conclude the restriction enzyme has worked on my DNA? So I do a cross verification step, that is gel electrophoresis. So what is gel electrophoresis? So I take a gel made up of agarose. So agarose is a seaweed, right? So I take a gel made up of agarose. So it is commercially available. So I take the agarose. So then I add it in the appropriate proportions and I solidify it. So when I heat it and I solidify it, it becomes like a semi-solid gel. So it ideally looks like this. But here I make depressions, that is called as wells. So I load my sample here. So this is called as wells. So once I load my sample, before loading the sample or while making the gel, to the gel I add ETBr. So ETBr is ethidium bromide. So ethidium bromide enables you to see the bands under UV light. So ethidium bromide is an intercalating agent. So it helps you to view the bands under UV light. Fine. And to the sample, you mix the sample sorry. So this is my DNA sample. So this is this DNA plus my restriction enzyme. So now to this sample, I'm going to check if my restriction enzyme has worked or not. To this sample, I add a tracking dye, that is bromophenol blue. So I load it in the wells, and when I load it, I give an electric field. So this electric field is from 50 millivolt to 70 millivolt. So now the particles move from, or the fragments move from negative end to the positive end. Because your DNA is negative, so this side it will be negative end and this side it will be positive end. Okay, so the fragments move from the negative end to the positive end, and smaller fragments move faster. So this is the principle that your gel electrophoresis follows. So it is based on the size. Smaller the size, the quicker it moves. Larger the size, it takes much time, right? Okay. So now, how do you know which is what size? So for that, you add what is called as a reference or a ladder. So this reference or ladder is also commercially available, and it lets you know what is the size of each of the fragment, and accordingly you can find your gene of interest, right? So from the gel you take it back in a vial. Okay, so this step is called as elution step, for which you use elution buffers, etc. to dissolve the gel and get only the fragment of your DNA. Done? Okay. Now, the next thing that you should understand is now my gene of interest is ready. Okay, so my gene of interest is ready. So now I have to put this in a vector. A vector or a plasmid is double-stranded extracellular material, right? Extra chromosomal material. So this is double-stranded. Now, to put this inside, I have to cut open this. So I use the same restriction enzyme that is used to cut open my gene of interest. So I cut this open using the restriction enzyme. So now what will happen? So I have to join my vector and my gene of interest like this. So while joining these two, the process is called as ligation. So ligation is done by ligase enzyme. So ligase is a family of enzyme that joins two bonds, okay? So it joins the gene of interest with the plasmid. Because we have cut it with the same restriction enzyme, it has compactable sticky ends or blunt ends, okay? So now, how do I cross verify this ligation has happened or not? So today we're going to talk about the cross verification step as well as the features of a plasmid. So we are going to talk about the features of a plasmid. So we will talk about different types of plasmid to some extent. And how do you cross verify? So the checkpoint step or the cross verification step is using antibiotic resistance. So this is one. The second one is insertional inactivation. Fine? Yes? No, these two are not done. So whatever I explained before was done. I was just revising. Okay, W? So now we talk about cloning vectors. So what are the features of a cloning vector, or how do I define a cloning vector? So number one, DNA is used as a carrier for transferring a fragment of foreign DNA into a suitable host through a vehicle or a vector DNA. The one which carries the DNA into a host, that is a vector or a plasmid. Alright, so we call it as a vehicle or a vector DNA. So what can be used as a vector? So your plasmids can be used as a vector. So plasmids are naturally occurring extra chromosomal double-stranded DNA. Yes. And bacteriophage. So bacteriophage are viruses that infect bacteria. Fine. So these bacteriophages, they have the ability to replicate within the bacterial cells, because a virus will also know how to replicate within a living cell. So we are exploiting that property to transfer a foreign DNA. Alright, so plasmids and bacteriophages have the ability to replicate within the bacterial cells independent of the control of the chromosomal DNA. So like I told you in the start, so if this is a bacterial cell has the genomic DNA, the DNA that codes for proteins essential for its survival. Apart from that, it has extracellular circular chromosomal DNA. So these are plasmids. So these are extra. They are not needed, but they are naturally present. Okay. So these plasmids, they replicate because ultimately it is DNA, right? Your DNA has the ability to replicate. That is why it is your genetic material. So your plasmids replicate independent of your genomic DNA. Right? So this is the next point. So bacteriophage has high number per cell, so which means it produces very high copy numbers. So if I have many number of bacteriophages, for example, what is a bacteriophage? A virus which is getting inserted into a bacteria is a bacteriophage, right? So instead of the virus genetic material, if I replace it with my gene of interest, now it acts as a plasmid. So virus enters inside the body, it only integrates this genetic material with the genetic material of the bacteria and the entire thing dies. Okay, so this DNA or the RNA gets integrated with the genomic DNA and it continues replication. Okay, so if I have many copies of the plasmid, that is called as copy number. In how many copies the plasmid or the bacteriophage is present, that is called as copy number. No, not Xerox copy. So if I have 10 plasmids present inside a particular bacterium, so I say the copy number of this plasmid is 10. So for example, if I allow one particular division of a bacteria, and if it gives me in one division, it gives me 30 copies of the plasmid. So that is my copy number that I am talking about. How many copies it can replicate and produce, alright? So plasmids, some have 1 to 2 copies per cell, but bacteriophages can replicate and produce many copies of itself, alright? So some plasmids have 15 to 100 copies per cell. So depending on how quickly they can replicate, that is very important. So copy number is very important. Alright. So this is the introduction to cloning vectors. So now let's move further. This is how a typical cloning vector looks. So what are the regions that it has? First, a cloning vector should have an origin of replication. So this is your origin of replication. So it should have an ori site because plasmids are capable of replication, right? So it should have an ori site, and this ori site should be flanked by a promoter sequence in the front, because a promoter is the one which stimulates the ori site to start the replication. So apart from that, it should have a restriction site, because if I want to cut open the plasmid, I look for the restriction site so that I can use my restriction enzyme. So it should have some restriction site, and it should have a selection marker. This selection marker is used for the cross verification step. So this selection marker is used for the cross verification step. Alright. Okay, so here we have, so this is how a normal plasmid should be. Can we create our own vector? Yes, that is possible because we know what are the things that is present inside a plasmid, right? So now you can recreate. So we cannot make the pizzas exactly supplied by Domino's, but we know what are the ingredients and we can recreate a miniature version of the Domino's pizza, right? So now that we know what are the features that is present in a cloning vector, we can have the same features and we can also recreate a plasmid, okay? So now here we go. So one of the first plasmid that was recreated or that was artificially engineered was your PBr322. Another plasmid which was engineered was your PUC19. Okay. So now what does this stands for? P stands for plasmid. B and R stands for the scientists who discovered them, Bolivier and Roderick, and 322 is the number or according to them whatever they wanted. Here P stands for plasmid, UC stands for University of California because that was where this first plasmid was constructed. Okay, yes. So that is the name because they have artificially engineered it. Okay, W? Right. So now what are the regions that is present here? Let's see. So the first one is the oocyte. So oocyte is needed because that is the one where your origin of replication begins, right? So very close to the oocyte you have a promoter, but promoter is not significantly mentioned here, and it has a ROP enzyme which is essential for recombination. Sorry, it is essential for replication. Okay, some extra enzyme so that replication happens properly. Apart from that, if you see, it has restriction site. This PVU2, BAMH1, HIND2, HIND3 and ECOR1 are the restriction sites. These are restriction enzymes, right? Yes, so restriction enzymes are mentioned here. So the restriction site or the recognition site for these enzymes are present at these points. A vector should have restriction sites, right? So only then I can use my molecular scissors to cut open the plasmid. So now if I am using this plasmid PBr322, so I have various options. So I can use PVU2 plus enzyme, BAMH1 enzyme, HIND3 enzyme or ECOR1 enzyme, right? So I have many options. But apart from that, if you see, there is something called as AMP-R and TET-R. So this is your selection marker that I was talking about. So what does AMP stand for? AMP stands for ampicillin. So what is ampicillin? Ampicillin is an antibiotic. So when do you take antibiotic? So we often eat antibiotic when we are not well, no? So why do you take antibiotic? Yes, anyone? Yes, W, why do you take antibiotic? Yes, any idea? Yes, why do we take antibiotic? Okay, so when you have an infection by bacteria, to kill the bacteria or to fight against the bacteria, we take antibiotics, right? So one such antibiotic is ampicillin, but here it is written as AMP-R. So what does this R mean? R means resistance. So this is a gene that codes for ampicillin resistance. So understand, this is a plasmid. I am going to insert this plasmid inside a bacteria, right? Because I have to produce many copies first. That is called as cloning, right? So gene cloning or cloning means to produce many copies, which I explained in the first lecture. So ultimately, I am going to put this inside bacteria, right? So sometimes if I have these antibodies or if I have these antibiotics, these will kill my bacteria. But if I have my resistance gene, it will survive in that condition. So that is also the reason antibiotic resistance is present. So today, whichever antibiotic you take, bacteria is becoming much faster, much smarter because it is developing resistance against that particular antibiotic. Also, why is the function of these antibiotics? Let me tell you. So TET-R stands for tetracycline, which is also another antibiotic. Okay. Some suppose if I cut open my plasmid at the 𝐵𝐴𝐻1 site. So I am cutting open the plasmid here. So it cuts like this. So here I insert my gene of interest. So my 𝐷𝐻 gene is lost, right? So now if I have to cross verify if my gene of interest has properly fitted into my plasmid, I will grow the bacteria in a tetracycline medium and check. If the bacteria survives, it means it has this 𝐷𝐻 gene. So which means ligation has not happened. But if my bacteria and the plasmid dies in tetracycline because it does not have this resistance gene, so I am very sure that yes, ligation has happened. So this is how we cross verify. So once again, so I have a plasmid. So this is my plasmid. I draw it according to my convenience. So this is my oocyte. So this is my 𝐕𝐵 enzyme which helps in recombination. So here I have my ampicillin resistance gene, AMP-R, R stands for resistance, and here I have my tetracycline resistance gene, so 𝐷𝐻, right? So here I have my 𝐵𝐴𝐻1 site. So I cut using the 𝐵𝐴𝐻1 restriction enzyme. So when I cut it like this, the tetracycline gene is lost. Right? So tetracycline gene is chopped off. So now what do I get? So I don't have the tetracycline gene, but I have all the other genes. So I have AMP-R, so I have oocyte, and I have the 𝐕𝐵 gene. Right? So now I have my gene of interest waiting here. So I am trying to insert my gene of interest here. So that is called as ligation. So now if the ligation has happened, so say this is my gene of interest. So ligation has happened. So here I have the remaining things intact. So AMP-R, so here I have the oocyte and here I have the 𝐕𝐵 gene. Now, how do I cross verify that it has ligated? So now I will grow the plasmid in an ampicillin medium, in a tetracycline medium. So if I grow the plasmid in a tetracycline medium, it should actually die because I have removed this tetracycline resistance gene. If the bacteria dies, I am very sure ligation has happened. But if it lives, then something wrong has happened. It has not integrated in the right place. So that is how I cross verify. Alright. So now going one by one, features of cloning vector. So number one, origin of replication, which is called as the oocyte. So oocyte is very, very important because it decides the copy number of a plasmid. So this decides the copy number of a plasmid, and copy number is very important. So you will know how many clones you will get. So always when you're doing a life science work, you make copies and you store it. Suppose if I want to take a very, very ancient gene and do a cloning, so every time I cannot go back to the first source, right? So I have to make copies and keep. So I make clones and keep. To make clones, I have to know the copy number and also understand if I have one plasmid, the molecular volume will be very less. Anyways, it is all in very small quantities. So I need some more volume, right? So only then I can at least see the solution. So that is also the reason we produce many copies to prevent failure, to prevent the probability of something getting mixed. Okay. So origin of replication should be strong and it has to be flanked by a promoter sequence, and that decides the copy number of a plasmid. Alright. So selectable marker. A selectable marker is very important. That is your antibiotics, that is AMP-R and 𝐷𝐻 that we were talking about. So transformation, what is transformation? So now if I have my plasmid with my gene of interest here, so now I call this as recombined plasmid. So this is also called as transformed plasmid. So transformed means the ability or to change its original form. So transformation is change from one form, right? So it has changed from its original form to the new form. So a selection marker is used to check the number of transformants, to check if the plasmid has transformed. It's a procedure when a piece of DNA is introduced into the host bacterium medium. So now I am introducing the gene of interest into the plasmid, right? So genes encoding resistance to antibiotic like ampicillin resistance, chloramphenicol resistance, tetracycline or kanamycin. So these are the 4 antibiotics that are commonly being used. Alright. So normal E. coli cells do not have any resistance to these antibiotics. So in that case, you'll have to build an artificial plasmid with a selection marker so that you will be able to cross-check them. Alright. So fine. So now I can use either ampicillin site or tetracycline site to find out if my cloning or ligation has happened or not. But let me also tell you, to have your tetracycline and ampicillin, it is very tough because I have to do plating. So how do I do plating? Is you have what is called as Petri plates, okay? Yes, you have to. That is very important, W. Okay. So you have plates in lab. So I am doing all this experiment in lab, right? So I have plates like this that is called as a Petri dish. So on the Petri dish, I have a media which is the food for my bacteria. Okay. So this media is agar plate or agarose plate. So on this the bacteria grows. So if I have to give ampicillin or tetracycline, I add it in the media. So if I am adding tetracycline to this, the plasmids which have the TET-R gene only will survive. But our transformant should not have this TET-R gene, right? Because so that is when we know it has transformed. But plating, doing all this is a very, very tiring job. Because in life science, you always have to do replicates to make sure your answer is right. So it is a very tiring job and it can take the entire day. So we have come up with an alternative. So that alternative is called as insertional inactivation. So what is insertional inactivation? Very simple. So they have taken a plasmid. Done. So they have the ORI site, no change. So ORI site is flanked by a promoter. And here you have a gene. Say for example, this is a gene that codes for the enzyme beta-galactosidase. So we know what does beta-galactosidase do. We have read in regulation of gene expression in molecular basis, right? So beta-galactosidase breaks your lactose into glucose plus galactose, right? So now in this gene, I make sure I have restriction site. So I take off this gene and insert my gene of interest. So here I have a gene that codes for beta-galactosidase. Also here I have restriction site for HIND2 for example. So now I am cutting off this gene and inserting my gene of interest. So here instead of the gene that is coding for beta-galactosidase, I have my gene of interest. Okay. But now how do I cross verify? So, even if I give lactose, I will not know whether beta-galactosidase is breaking it down into glucose or galactose because again it is not visible. But when I give a substance called as X-gal, beta-galactosidase will break X-gal to give me a blue colored product. So now to cross verify my ligation, I will give X-gal from top. Now if the bacterial plate turns blue, it means beta-galactosidase gene is there because that gene is coding for that protein and hence the blue color is coming. But if I do not have blue color, if I have only white color, which means my ligation has happened. So here white color refers to the transformed ones, which we call it as transformants, or the blue colored ones refers to the non-transformants. Done? Did you understand? Is it simple? It's very easy, it's very interesting because we are playing at the genetic level, right? Any doubt? Any doubt? Clear? So that is all that I want to say. So again, okay, so these are extra cloning vectors which we'll do in the next class. So I hope first half you have understood. So now I have a list of cloning vectors that you can use for plants and a list of cloning vectors that you can use for animals, which we'll do in the next class. Okay, so I guess I have covered the important things in a small bits and pieces, but I hope you guys understand, right? If you don't, please ask me. I'm ready to explain once again. So alright, so that's all for this evening. We'll stop. So we'll continue on Friday with part 5 of biotechnology. So please join. We'll finish off this chapter because only one bit is there, that is competent host. Alright, so thank you so much for joining me this evening. So looking forward to meeting you on Friday. So until then, this is me signing off. So take care and thank you and bye.